Okay, I want to start by thanking the organizers for inviting me. Okay, so I'll be talking about our more recent studies on caloric restriction, which focus on age-related hearing loss, but I'm also going to give a little bit of background on the work we've done over the years relating to mitochondria and aging as a whole, and the connection to caloric restriction. So I was going to give a short introduction on caloric restriction, but this one is not working. Okay. Hello? Working? Okay. Finally. Yeah. So I'm not going to spend a lot of time with it since Jamie Barger covered this. But of course, the basic finding that we're trying to understand at the molecular level is how is it that caloric restriction has such a profound effect on lifespan, as shown in this slide. This is basically data plotted from the previous slide from a study from Rick Weindruck many years ago, where what you can see is that the change in lifespan is actually directly related, or more directly, inversely related to the caloric intake, and there's a linear relationship. And what Rick Weindruck has argued over the years is that this probably suggests a role for energy metabolism in the effects of caloric restriction. Now, caloric restriction, of course, impacts many health parameters. The calorie-restricted animals don't just live longer, they're biologically younger. But despite 7 decades of study, the mechanisms by which caloric restriction extends lifespan are not known or just beginning to be known. There's an inverse association between CR and longevity, suggesting a role for metabolism. CR does lower oxidative damage, which it's not a universal finding, but I would say in general that's what one sees, which links it to the free radical theory of aging as well. One area that has interested us a lot is that CR prevents age-related mitochondrial DNA mutations and age-related mitochondrial dysfunction. So here's the same slide that Jamie showed yesterday, showing that the mice on CR on the left at 40 months of age look quite a lot younger than their ad-lib fed siblings. Now, if we look at the effects of genetic manipulations on mitochondrial function, there's a few very interesting studies. This is one that we did a few years ago, where we basically genetically engineered the mitochondrial DNA polymerase gamma. This is the only mitochondrial DNA polymerase. We engineered it so that its exonuclease domain was defective, so that mice now had very high levels of mitochondrial DNA mutations. And what we observed in that case was a very dramatic acceleration of age-related phenotypes. The model we came up to explain these, based on a number of studies, was that as a result of these mitochondrial DNA replication errors, mitochondrial DNA mutations accumulate. That leads to electron system dysfunction. We have many lines of evidence that this activates apoptosis. And then there's loss of, in tissues that undergo high levels of proliferation, such as the lining of the gut, thymus, and some other tissues, skin, the accumulation of these mutations leads to loss of stem cells, and these are irreplaceable, and once a certain number of these cells, a certain percentage of these cells are lost, the tissue no longer has the ability to regenerate, then you get tissue dysfunction and aging. And the post-mitotic tissues is probably not so much loss of irreplaceable stem cells. It's more a kind of an overall loss of cells, and I'll show that for the particular case of age-related hearing loss. This is the summary of phenotypes of everything that we see in these mitochondrial mutator mice. You can see there's weight loss, muscle wasting, decreased bone density. And we observed age-related hearing loss and decided to pursue this a bit further based on our findings here. So, age-related hearing loss, also known as presbycusis, is a general feature of mammalian aging and is the most common sensory disorder in the elderly. It affects nearly half the human population between 50 and 90 years of age. And the reason is because the mammalian inner ear has a very limited regenerative capacity, and its function depends largely on long-lived post-mitotic cells. Just to give you a general schematic of what we're talking about, the organ that seems to degenerate with age quite dramatically is the inner ear, and particularly cochlea. And in the cochlea, you have these hair cells, which are shown here, that contact this tectorial membrane. The hair cells respond to different frequencies, and they connect to the brain through these spiral ganglion neurons. There's a part of the inner ear called the stria vascularis, which produces this endolymph, which is high in potassium. And that endolymph is required for the operation of this system, basically. And these cells also degenerate with aging. So with aging, you have loss of these cells, these, and these. And in humans, it seems to be a mixed phenotype, meaning there's loss of all three cells, all three cell compartments. There are cases where there's a more pronounced loss of one of the compartments that presumably would lead to a more accelerated age-related hearing loss. Now, hair cells are metabolically active and very rich in mitochondria. This is EM picture, as you can see, and you can see all the mitochondria there. There's quite a lot of them. And makes sense because of the high metabolic demands in these cells. The other thing that makes age-related hearing loss very interesting to us is the fact that you can easily measure it in mice. You can anesthetize mice, connect these microphones to them, and use this system where you produce sound in their ears and you measure the ABR threshold. Basically, you produce sound of increasing intensity at different frequencies, and you determine, what's the lower limit of hearing for these animals. And that, of course, changes with age as the hearing declines, and this is illustrated here. These are actually the mice that carry the mitochondrial DNA mutation. So what you can see is that the thresholds for different frequencies are similar between wild type and these mutant mice, which we call D257A because of the name of the mutation. This is in young mice. In old mice, they separate, and old here is only 10 to 11 months because these mice age quite quickly. And what you can see is there's a big separation between the wild types, which are still hearing pretty well, and the old mice for which the thresholds have raised. Essentially what it means is you have to make more noise for them to be able to detect the sound. At the cellular level, what we see is a loss of both the hair cells and the spiral ganglion neurons. So this is a young animal. Well, it's a wild-type animal, 9 months of age, relatively young. Here's the D257A mutant. You can see, it's hard to see the loss of hair cells, but for the spiral ganglion neurons, it's pretty obvious what's going on here. One of the interesting findings we made, in fact, we basically repeated it since it had been reported by a few other groups in the literature, but we did it with the black 6 mice, is that caloric restriction completely prevents this phenotype. So you've got 3 different frequencies here, 4, 8, and 16 hertz. And what you can see is that by middle age, there's a big increase in the thresholds, but if the mice are under caloric restriction, it's completely prevented. I said by middle age because these mice are essentially completely deaf by 15 months of age. So this is a really early onset phenotype. For most mouse strains, including Black 6, there are some strains that will live. Okay, so when we look at the histological assessment, what we see is that there's this loss of spiral ganglion neurons by middle age in the control animals, but it's completely prevented by caloric restriction. So the histology matches the hearing findings, the ABR thresholds. Now, how are these cells lost? Well, we suspected that it was through apoptosis, in part based on our work with the mitochondrial mutator mice, which had shown that mitochondrial dysfunction leads to apoptosis, and we tested this in the normal mice, and what we found was a big induction in apoptotic cells as determined by tunnel staining by middle age, and caloric restriction largely prevented that. So the idea is that caloric restriction somehow prevents this age-related apoptosis. So the next question was, how does it do it? So we did a DNA microarray experiment trying to understand which genes were either induced or downregulated by normal aging and caloric restriction. And this was some of the more interesting findings. So genes involved in apoptosis were downregulated by caloric restriction, and some genes related to mitochondrial function were upregulated by caloric restriction. So we came up with this general model where apoptosis induces the age-related hearing loss and caloric restriction suppresses it and induces mitochondrial genes. So here's the model. So with age, there's oxidative damage in the cochlea. Pro-apoptotic genes such as BAC and BAX, which are well known to play a role in mitochondrial apoptosis, go up. That induces cell death, mitochondrial apoptosis, and cell death, so you lose those critical cells. Now, BAC and BAX are very interesting genes. Mice deficient for these genes are viable, but mice that are deficient for both genes show developmental defects, so these are involved in apoptosis as related to development. BAC induces apoptosis in response to oxidative stress, and expression of BAX and BAC is induced by DNA damage through p53. So the critical experiment that we did was to obtain some BAC deficient mice and ask whether they undergo age-related hearing loss. So here what we have is the young mice, 5-month-old wild type and BAC deficient animals. They're young, they both hear quite well. Then with aging, you see the wild type mice have the higher thresholds, but the BAC deficient mice have a very significant prevention of age-related hearing loss. So this experiment basically shows that age-related hearing loss occurs through BAC. If you don't have BAC, you don't get age-related hearing loss. And that, again, matches very nicely the histological data. These are the young animals. They still have a lot of spiral ganglion neurons and hair cells, but at the older age, you can see how the wild-type mice look and the preservation of cells in the BAC deficient mice. This is just the tunnel staining showing, not surprisingly, that when you delete BAC, you basically prevent apoptosis, which goes up quite significantly by 15 months, but not in the BAC deficient animals. We also did some experiments with cochlear cultures using paraquat, a well-known oxidant. And what we did is we looked at both cochlear neurons, the whole cochlear cell extract, and cochlear neurons. And what we found was that essentially at all doses of paraquat, BAC deficient cells were protected. Now I want to quickly shift gears here to SIR3. So as I showed in one of the first slides, caloric restriction induced a number of genes involved in energy metabolism. One of them was SIRT3. So SIRT3 is the mitochondrial sirtuin, and it controls acetylation status to a large extent of mitochondrial proteins. It hasn't received as much attention as SIRT1, for which a lot more studies have been done. But SIRT3 is quite interesting because it connects the sirtuin story to mitochondria. So it's the major deacetylase, deacetylates isocitrate dehydrogenase, a major source of mitochondrial NADPH, which is required for regeneration of GSH. Its overexpression in cardiomyocytes protects against oxidative stress, and SIRT3 deficient mice show severe bioenergetic deficits, again confirming the central role of this sirtuin in mitochondrial metabolism. Again, the critical experiment here was to obtain some SIRT3 deficient mice and test whether caloric restriction can prevent age-related loss, hearing loss in these animals. And what we found was actually quite dramatic, which was that, again, with age, there's an increase in thresholds in normal mice. It's prevented by CR, but when we do the same experiment with SIRT3 deficient mice, there's no protection whatsoever. So the protection by CR, the complete prevention of age-related hearing loss, depends entirely on SIRT3, this mitochondrial sirtuin. Interestingly, we also had SIRT3 heterozygote mice. The homozygote mice were too sick, so we looked at the heterozygote mice. And what we found was that here the surprising observation was that age-related hearing loss in mice under a normal diet doesn't occur in SIRT1 deficient mice. So what that actually means is that you require a full gene dose of SIRT1 to be able to achieve age-related hearing loss. So whereas SIRT3 seems to prevent age-related hearing loss through caloric restriction, SIRT3 is actually required for it, independent of caloric restriction, SIRT1, I mean. So it's very surprising because SIRT1 has been proposed to be the main mediator of caloric restriction effects, but in this model it seems to have the opposite effect. We have to follow up on these studies. I mean, this is the only data we have, and it's such a surprising result that we certainly need to reproduce it. We also did some studies trying to understand how CR modulates age-related hearing loss through SIRT3, and we looked at the literature, especially some studies done by Raj Sohal several years ago, and what he had shown was that when you put animals in CR, there's a big increase in the GSH GSSG ratio. So it essentially doubles, and it depends on the tissue. In the inner ear, we see essentially a doubling. It's quite dramatic effect of CR, and we ask whether that happens in the SIRT3 deficient mice, and it doesn't. We also looked at liver and brain, and we found that this effect seems to be universal. So CR induces glutathione in multiple tissues, and it doesn't do so in SIRT3 deficient mice. Now, glutathione, of course, is the main cytosolic, water-soluble cytosolic antioxidant, so we think this is very important, and we think that the mechanism is that CR induces SIRT3, which in turn decreases IDH2 acetylation, leading to higher IDH2 activity, higher mitochondrial NADPH, and improved glutathione GSH-GSSG ratio. We also show that SIRT3 and IDH2 physically associate. This just summarizes what I just said. I'm going to try to go quickly. This just shows that with CR, we get the increase in IDH2 activity. There is no effect in the SIRT3 deficient mice. The same for NADPH levels. It's a small effect, but it doesn't occur in SIRT3 deficient mice. This just shows that SIRT3 deacetylates IDH2, and we think that's the biochemical metabolism underlying these effects on NADPH levels. You can see that there's a quite dramatic change in relative IDH2 acetylation with CR, and that under SIRT3 you don't see that. There's also effects on DNA damage, so CR lowers DNA damage. We don't see that in the SIRT3 deficient mice. So the overall finding is that many of the age-related phenotypes seem to be dependent on mitochondrial SIRT3. I think I'm going to close with this slide. So our basic model is that aging induces mitochondrial free radicals that leads to nuclear DNA damage, which in turn activates BAC-mediated apoptosis. Now, I say nuclear DNA damage because BAC is expressed, is encoded in nuclear DNA, not in mitochondrial DNA, and it's known that it's nuclear damage that induces BAC expression, but it could be damage induced by mitochondrial ROS. That in turn leads to spiral ganglion neuron and hair cell loss. I'm going to skip these last slides. Just want to thank my postdoc, Shinichi Someya. He's now an assistant professor at the University of Florida. He did the great majority of the work I described today. He specializes on age-related hearing loss, so it was really productive to have him as a postdoc. We've had many collaborations in this work, including Rick Winder, Christian Leuenberg, and Peter Rabinovich. Thank you.
Now, the discussion is open. There are questions. I have a more general question. You said that you mentioned that there is the action potential in the hearing actually is due because the high potassium that is outside. It's one of the few cases that you find in physiology. I was wondering if you know if you have also, since you have a high potassium outside is due to active pumps, and if you modify mitochondria, you maybe activate also ATP. So I was wondering if you there is also maybe you lose the capacity of hearing just because also you don't actually have a good action potential.
Yeah, so it's well known that at least in mice, for some strains of mice, there is loss of that endocochlear potential. And it's thought to be due to loss of cells in the stria, the cells that probably produce the changes in currents there. But I'm not a hearing physiologist, so I don't really understand very well the biochemical basis of the production of the endocochlear potential. I just know that it is altered with aging. We never measured it, though. It's quite complicated.
So there are other mechanisms for generating NADPH in mitochondria. And so it seems to me that it shouldn't be all or nothing for isocitric dehydrogenase. Can you comment on that?
Yes. So I think IDH2, if I remember correctly, produces about half of the NADPH in the mitochondria. So there are other mechanisms, and we haven't examined the effects of SIRT3 on those. It may be that even a partial lowering of or increase in NADPH already leads to enough of a boost of the antioxidant system that maybe prevents the age-related hearing loss. I mean, when we say prevent, we mean by 15 months of age. I mean, if we age these mice to 20 or 25 months of age, maybe they'll show it. So we haven't taken it that far.
Thomas, I enjoyed a lot your talk because you stressed the importance of SIRT3. And indeed, I would like to remember that some years ago, a few years ago, in a large study on more than 1,000 centenarians, we looked at polymorphism of chromosome 11, 15.5, and the SIRT3 polymorphism emerged as the most significant correlated with longevity in humans. So I think that you are right, that SIRT3 maybe is one of the most important genes in longevity.
Follow a little bit of Lester's question. I think it's far more than 50% of the NADPH which is produced in mitochondria is not IDH2, but the nicotinamide.
Yes, the NA.
Hydrogenase. But one of the targets of SIRT3, the deacetylase activity, is actually alpha-ketoglutarate dehydrogenase. That means that the impairment that you are seeing is not on the redox component, but essentially on the production of NADH by the ketoglutarate dehydrogenase. That means that this is an energy deficit associated with the redox component. Yeah. I think if one would evaluate the energy transducing capacity of mitochondria, it will be very, very diminished, and again, because of impairment of the ketoglutarate dehydrogenase, not because of the IDH2s. What you want to do is to associate caloric restriction, circular activity, and oxidative stress. But I think that the energy component here probably is more important than the redox one.
Yeah, so as far as linking oxidative stress, these studies were a bit descriptive in the sense we're correlating. But we have shown, though, that if we overexpress a mitochondrial catalase transgene in mitochondria, it prevents age-related hearing loss. So oxidative stress, and also if you feed in some antioxidants supplements, so we fed more than 50 to these mice, and we found that CoQ10 and lipoic acid prevented age-related hearing loss. So there is some link to oxidative stress, it seems, unless these are acting by...
But what I'm trying to say, that cannot be viewed as an isolated event which is only related to oxidative stress.
Yes, I agree. And in fact, as I mentioned in the introduction, another group has published that these mice do have very low ATP levels, so there's clearly an energy deficit.
Okay, last question.
Did you check what happens when the bucket is
There's the microphone.
Did you
Back,
check what happens when the bucket is overexpressed?
yeah, no, we don't have the back transgenic model. So.
Okay.
And any data on bugs?
Oh, yeah, so I forgot to say, we tested BACs and it doesn't have any effect. So BACs deficient mice show age-related hearing loss just like the wild type. So for the cochlea, BAC is the important player.